So here we're going to learn about the Lac Operon and also see a simulation on exactly how it works to get a better understanding for it. So first off, the Lac Operon, Lac stands for lactose, and this is an example of a sequence of genes. So an operon in general is a sequence of genes, may be found in many bacteria that encodes for enzymes needed to break down lactose to glucose and galactose, so sugars can be utilized. Studies on the Lac Operon have been important for genetic research. The lactose receptor proteins control the transcription of the Lac Operon. Now this operon, again, it is in bacteria. So I do show the glasses of milk here, and you think if you drink a glass of milk, you're activating your own Lac Operon. Not necessarily the case, but this is give a good example here of taking the lactose and with the lactase enzyme and breaking it down into galactose and glucose, which can be absorbed by the body and other organisms. So the Lac Operon, the lactose operon to be the full term here. An operon is a segment of DNA that contains a cluster of genes that are transcribed as a unit and are involved in lactose metabolism. Lac is short for lactose, the disaccharide composed of galactose and glucose that is found in milk and other products. But as I stressed before, note that operons occur primarily in prokaryotes. Keep in mind you are a eukaryotic organism. So while operons do occur mostly in prokaryotes, this is a simulation that does provide a good model for gene expression. So take it for what it's worth in that regards. Now when we're looking at the Lac Operon, it's repressed when no lactose is present. So the cell doesn't want to make this enzyme if there's nothing to break down. So in the absence of lactose, the Lac Repressor binds to the operator. That repressor here is going to prevent that gene from being made. The operator here is kind of like the light switch that says go or don't go. So by binding a repressor, it's going to prevent anything from downstream of that from being expressed. So RNA polymerase cannot access the promoter, therefore the Lac Operon is shut down. In this example, it's important to shut down if there's no lactose present and a high glucose. Glucose is a favored energy source. Now in low glucose and lactose not available, again, the Lac gene is also not going to be expressed. Now Lac Operon is activated when lactose is present. So in this case now we have lactose being available and we have low amounts of glucose. So in the presence of lactose, the metabolite of the lactose is called allolactose, binds to the repressor, and this induces a change in the shape of the repressor, making it fall off the operator. This now will expose the operator so that RNA polymerase can now bind to the promoter, and essentially this gene or these genes now are turned on and these will be made. And it's important for the cell to make them for the bacteria cell because now lactose is available. This gives you a couple scenarios here. We talked about lactose being available. Yes, we want to have that gene strongly expressed. In these two following cases, we have lactose not being available, so therefore they are repressed. They're repressed for different reasons. In this case, we have high glucose, so the cell's still getting energy. Here's not a lot of energy available regardless, but still not a lot of lactose, or lactose being unavailable. Those genes will not be expressed. Here we have high glucose and lactose being available, so there will be some, we call it low basal level gene expression, because as I mentioned, glucose is preferred, but in the case where we have low glucose and lactose being available, this is going to be strongly expressed. This is simply the bacteria cell producing the genes that ultimately will produce the proteins that are most efficient for its current conditions. Now this is that Operon simulation. I give you a link to it right here. Another video, you could take a look at it, but I'm kind of go through the simulation now so you get an idea of exactly how this works. Okay, so now we're here looking at the simulation. We see our legend here, and we have our ability to add lactose to the situation. So here we have some breaks in our genetic code. So I want to add that promoter region, so that Lac promoter region. But if we add just the promoter region with no gene, this is our RNA polymerase binding to that, but it can't make the gene. So here now I have the gene. When I attach the gene there and polymerase goes through, now it has that specific sequence to replicate. That gene will go through the process of translation to form what this is going to be is the repressor. So here's our Lac Z gene. If I insert the gene, it's all well and good. Now we have a complete genome. However, we don't have an area for polymerase to start. So it's very important that you add the promoter. We also want to add that very important operator. And you'll notice that operator fits the repressor here. So we're not making the Lac Z gene. We're not making the lactase enzyme. Why? There's no lactose present. So if I add some lactose, you can see our disaccharide here. When this comes through and binds to the repressor, it changes its shape. It removes that repressor. So I add a little more lactose. Now that repressor is removed, now we can make that Lac-Z gene, which is only going to go through the process of translation to form our Lac-Z enzyme. This enzyme can go through and break down the disaccharide into galactose and glucose. Now you'll notice that I haven't added any more lactose, and we've broken down almost one more piece here. We've broken down all of the lactose that was present, and now the repressor is bound here and kind of stuck here. It's going to prevent the enzyme from being made. It's this very efficient way for the cell to go through and not waste its energy. Now, if this bacteria cell goes through and for whatever reason comes into an environment that's very high in lactose, see if I flood the area with lactose, now what we're having is we need a lot of these enzymes to be made. So all these repressors are being bound, their shape is changed, they can't bind here to prevent the making of that enzyme. You're now going to see a lot of enzymes being made and produced. And it's very important those enzymes are made and produced because we've got a lot of this lactose present. We have off the charts amounts of lactose. But going through the process here, we can see the enzyme's doing its job, multiple times breaking down the lactose, and those lactase enzymes doing their job, going through, breaking everything down. Still making more enzymes because, again, those repressors are bound. They are a different shape. They can't block that gene, which is important for the cell. High degree of lactose being present. We want a lot of those enzymes present as well. That's helping this breakdown process. And this will continue until we notice that there is basically not a lot of lactose remaining. Therefore, we have no need to produce the lactase enzyme. The repressors will come back through eventually, bind to the repressor, and reduce the amount of enzymes being made until the events change where there will be some more lactose present. You can see now we're overrun with enzymes, but the repressor is binding, preventing any new ones from being made. However, if we take this one more step further, look at lactose in transport. We have the same situation, but here we have the cell membrane. So we have lactose outside the cell. We have inside our cell a repressor being made. Let me add our Lac-Z gene. Let me add our promoter like we did and our operator. Operator right now, going to allow that Lac-Z gene to be made. We see our repressor being bound. Now that gene that allowed to be made, it didn't have the proper operator. We have some enzyme here. Even though there's lactose present, it is separated by the cell membrane. So there's one more gene here involved with this operator. It's called the Lac-Y gene. What this Lac-Y gene occurs is allows this kind of intermembrane protein to be made. Now you notice right now it's being blocked, but these repressors can't bind forever. So eventually it detaches, another one's going to come in right behind it. But now what's going to happen is we're now having the Lac-Z gene made to break down lactose. We're also having this Lac-Y gene made. This protein is ultimately going to go, this green protein, is going to ultimately go to the cell and allow the passage of that lactose from the outside of the cell to the inside so that enzyme can work. Now you'll notice the same thing. I add the enzyme, it's going through, it's being transported, now it's being broken down. And we're very efficiently using our enzymes. If I stop adding the lactose, we have no reason to produce any more of the Lac-Z or the lactase enzymes. We could see that that repressor is bound, but now one more time we've got changed its conformational shape. We're going to make another round of enzymes, which will break down these last bit of lactose that is present inside the cell. Now these got in the cell, and those proteins on the outside of the cell are still present. If we cause a mutation, so for example, if I take the Lac-I gene and I mutate it and basically remove it, we're no longer making any repressors. If we no longer make repressors, that's going to cause some issues because when these finally degrade, the cell is going to keep making these proteins even though there is no lactose present. So this is important for gene regulation, that we don't want the cell to be wasting energy making all these proteins. We want to make sure it's efficient, producing when it needs to, and hopefully this Lac Operon gives you kind of that idea of just how important gene regulation is, and some of the, hopefully a simplified breakdown of the complexities of some of the steps involved. Here where we have a mutation in the kind of repressor protein being made, now we have all these extra enzymes being made when there is no lactose. So the cell right now is wasting energy. We want to make sure we have all of our genes properly operating with functional promoters to allow the cell to be efficient with its energy usage. Okay, having gone through the simulation now, hopefully that was helpful for you. And if you go through, I got a couple questions here. So if you felt like you understood it, why do genes require promoters? What's the purpose of an operator? A couple other questions here. I try to carry through the simulation that I did. And you've got a couple other questions here to consider. You might want to pause the video, take a look at, make sure you can answer so you can determine your level of understanding when it comes to operators in regards to bacteria being able to break down lactose.